Another day, another story. Deoxyribonucleic acid, better known as DNA, is crucial to life on Earth. The questions and answers in this list are taken from the top questions sections of the articles on DNA, recombinant DNA, and chloroplast, where you can find more questions answered. Welcome to Tabo Eminent Channel. What is DNA made of? Structure of DNA molecule. Genetics, heredity, philosophy of science, nitrogenous bases, nucleotides, genetic code, biology, double helix, deoxyribonucleic acid, chromosomal, biochemistry, helical, biochemical, hereditary, biological, anatomy. DNA, or deoxyribonucleic acid, is a molecule that contains the genetic instructions for the development, functioning, growth, and reproduction of all known living organisms and many viruses. It consists of two long strands arranged in a double helix structure, composed of simpler molecules called nucleotides. Each nucleotide in DNA contains a sugar molecule, deoxyribose, a phosphate group, and one of four nitrogenous bases: adenine, A, cytosine, C, guanine, G, or thymine, T. The sequence of these bases along the DNA strand forms the genetic code, which determines an organism's traits. DNA replication is the process by which DNA makes copies of itself when cells divide, ensuring that each daughter cell receives an identical copy of the genetic information. This process is essential for growth, repair, and reproduction in living organisms. DNA is also subject to mutations, which are changes in the sequence of nucleotides. Mutations can occur spontaneously or be induced by various factors such as radiation, chemicals, or errors during DNA replication. Mutations can have a range of effects, from causing genetic disorders to contributing to evolutionary change. The discovery of the structure of DNA in 1953 by James Watson and Francis Crick is considered one of the most significant achievements in the field of biology, paving the way for advancements in genetics, molecular biology, and biotechnology. The human genome is made up of approximately 3 billion base pairs of deoxyribonucleic acid, DNA. The bases of DNA are adenine, A, thymine, T, guanine, G, and cytosine, C. DNA is made of nucleotides. A nucleotide has two components: a backbone, made from the sugar deoxyribose and phosphate groups, and nitrogenous bases, known as cytosine, C, thymine, T, adenine, A, and guanine, G. Genetic code is formed through different arrangements of the bases. What does DNA do? DNA in the cell nucleus carries a genetic code, which consists of sequences of adenine, A, thymine, T, guanine, G, and cytosine, C, Figure 1. RNA, which contains uracil, U, instead of thymine, carries the code to protein-making sites in the cell. To make RNA, DNA pairs its bases with those of the free nucleotides, Figure 2. Messenger RNA, mRNA, then travels to the ribosomes in the cell cytoplasm, where protein synthesis occurs, Figure 3. The base triplets of transfer RNA, tRNA, pair with those of mRNA and at the same time deposit their amino acids on the growing protein chain. Finally, the synthesized protein is released to perform its task in the cell or elsewhere in the body. DNA is an organic chemical that contains genetic information and instructions for protein synthesis. It is found in most cells of every organism. DNA is a key part of reproduction, in which genetic heredity occurs through the passing down of DNA from parent or parents to offspring. Who discovered the structure of DNA? The discovery of DNA's double helix structure is credited to the researchers James Watson and Francis Crick, who, with fellow researcher Maurice Wilkins, received a Nobel Prize in 1962 for their work. Many believe that Rosalind Franklin should also be given credit, since she made the revolutionary photo of DNA's double helix structure, which was used as evidence without her permission. The structure of DNA was discovered by James Watson and Francis Crick in 1953. They proposed the double helix model for the structure of DNA, based on X-ray diffraction images obtained by Rosalind Franklin and Maurice Wilkins, among other data. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Rosalind Franklin's contributions to the discovery were not recognized by the Nobel Committee, as the Nobel Prize is not awarded posthumously, and Franklin had passed away in 1958. Can you edit DNA? Gene editing today is mostly done through a technique called clustered regularly interspaced short palindromic repeats, CRISPR, adopted from a bacterial mechanism that can cut out specific sections in DNA. One use of CRISPR is the creation of genetically modified organism crops. Treating genetic disorders. Gene editing can be used to correct mutations that cause genetic diseases, offering potential treatments or cures for conditions such as sickle cell anemia, cystic fibrosis, and muscular dystrophy. Agricultural improvements. Gene editing can be used to create crops with improved traits such as increased yield, pest resistance, or nutritional content. Biomedical research. Gene editing can be used to study the function of specific genes by introducing mutations. What is recombinant DNA technology? Steps involved in the engineering of a recombinant DNA molecule. Biotechnology, DNA replication, DNA cloning, genetics, genetic engineering. Recombinant DNA technology is the joining together of DNA molecules from two different species. The recombined DNA molecule is inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. Since the focus of all genetics is the gene, the fundamental goal of laboratory geneticists is to isolate, characterize, and manipulate genes. Recombinant DNA technology is based primarily on two other technologies, cloning and DNA sequencing. Cloning is undertaken in order to obtain the clone of one particular gene or DNA sequence of interest. The next step after cloning is to find and isolate that clone among other members of the library, a large collection of clones. Do chloroplasts have DNA? Unlike most other organelles, chloroplasts in mitochondria have small circular chromosomes known as extranuclear DNA. Chloroplast DNA contains genes that are involved with aspects of photosynthesis and other chloroplast activities. It is thought that both chloroplasts and mitochondria are descended from free-living cyanobacteria, which could explain why they possess DNA that is distinct from the rest of the cell. Yes, chloroplasts have their own DNA. Chloroplasts are organelles found in plant cells and some other eukaryotic organisms, such as algae. They are responsible for photosynthesis, the process by which plants convert light energy into chemical energy in the form of glucose, using carbon dioxide and water. The DNA in chloroplasts is circular, similar to the DNA found in bacteria, and it encodes genes necessary for chloroplast function and photosynthesis. While chloroplasts have their own DNA, they also contain proteins encoded by nuclear DNA, which are imported into the chloroplast from the cytoplasm to support its function. The presence of DNA in chloroplasts is one piece of evidence supporting the endosymbiotic theory, which proposes that chloroplasts and mitochondria, another organelle with its own DNA, are the descendants of ancient prokaryotic cells that were engulfed by ancestral eukaryotic cells in a symbiotic relationship. Thanks for watching. Request you to subscribe the channel.